The effect of oxidative stress on the production of alpha-synuclein in C. elegans. Parkinson's disease is a very common and widespread disease in the United States of America. 1 out of every 100 people over 60 in the United States will suffer from Parkinson's disease. Over 60,000 people in the United States will be diagnosed with Parkinson's disease, with many other cases going undetected. Over 1 million people in the United States will suffer from Parkinson's disease. It is estimated that around 7 to 10 million people suffer from Parkinson's disease worldwide. Treatment for people with Parkinson's disease totals up to over $25 billion per year in the United States alone. Reactive oxygen species cause damage to an organism's body. This damage includes cell death and damage to an organism's DNA, lipid layer, and proteins. Cumulatively, ROS is called oxidative stress. Oxidative stress is the reason for aging in an organism. The more oxidative stress an organism produces, the faster the organism will age. Oxidative stress is hypothesized to be one of the main causes of Parkinson's disease and other neurodegenerative diseases. Alpha-synuclein is a protein that is abundant in the human brain. In many cases of Parkinson's disease, the alpha-synuclein gene produces either too much alpha-synuclein protein or an abnormal form of the protein, which is believed by some researchers to be toxic to brain cells and to also result in neuron dysfunction. It is thought that oxidative stress can cause an increase of production of alpha-synuclein and damage to the membrane that alpha-synuclein binds to. This oxidative stress allows more alpha-synuclein to bind to the membrane, which causes toxicity, which leads to cell death and neuron dysfunction. Since C. elegans have a short lifespan, experiments can be done with them in a short duration of time. C. elegans also reproduce very frequently and in large amounts, which help increase trial size, which will help make the standard deviations more accurate and smaller. Since C. elegans have a transparent body, yellow fluorescent protein can be used with image J and a fluorescent microscope to measure the concentration of alpha-synuclein in C. elegans.
Neurodegenerative disorders such as Parkinson's disease display accumulation of proteins called alpha-synuclein in the brain. This protein aggregates and forms insoluble fibrils called Lewy bodies, a typical hallmark of the disease. The normal function of alpha-synuclein is not clear, but there are evidences that this protein interacts directly with phospholipid membranes of vesicles and is responsible for vesicular transport across the synapses. Alpha-synuclein consists of a polar domain which interacts with the hydrophilic environment of the cytosol. It also has hydrophobic residues which is buried in the acyl chains of the lipid bilayer. These hydrophobic residues are also responsible for the toxicity of the protein. Also, there are positively charged lysine residues which separates these two domains and interacts directly with the anionic surface of the membrane. Under normal conditions, alpha-synuclein exists as native unfolded protein. In presence of lipid, it adopts alpha helical conformation. Next, the dimerization occurs on the membrane and it changes to beta sheet. This leads to the formation of beta oligomers, which results in protein aggregate deposit to form toxic Lewy bodies. The binding of alpha-synuclein to cholesterol and sphingomyelin containing vesicles could help in stabilization, possibly protecting them from premature fusion as a result of curvature stress. In healthy neurons, alpha-synuclein localizes to the synaptic vesicles and modulates vesicle pools at the synapse. In diseased conditions, alpha-synuclein increases lipid interactions and reduces curvature, leading to the clustering of vesicles in the reserve pool. This stalled vesicles creates high concentration of alpha-synuclein. Such enhancement of activity leads to synaptic dysfunction and damage to ER, mitochondria, and other organelles. These consequences of defects leads to toxic events leading to the pathophysiology of Parkinson's. In short, alpha-synuclein normally is a critical regulator of synaptic vesicle dynamics in vertebrates, whereas too much of its membrane interaction can lead to cellular dysfunction and development of Parkinson's.
Kongdong did an experiment on the effect of methyl blue on the production of the tau protein in C. elegans. The tau protein has been linked with many neurodegenerative diseases such as Parkinson's and Alzheimer's. There are two groups, a control group and a methyl blue group. It was found that in the methyl blue group that there was a significant reduction of the production of the tau protein compared to the control group. This shows that methyl blue could possibly reduce the production of the alpha-synuclein in C. elegans, since it reduced the production of tau protein in C. elegans. Gruber did an experiment on the effect of mitoQ on the improvement of longevity in C. elegans that have a strain which displays similar symptoms to Alzheimer's, along with wild-type C. elegans. It was found that mitoQ significantly improves the lifespan of C. elegans in both the Alzheimer groups and the wild-type groups. This shows that mitoQ does have an effect on the reduction of oxidative stress in C. elegans, and it can even slow neurotoxicity in C. elegans. It would be very interesting to see if mitoQ could decrease neuron toxicity and the production of alpha-synuclein in C. elegans with Parkinson's disease. Whitworth did an experiment on the effect of glutathione on the neurotoxicity in dopamine receptors in the brain of Drosophila. All groups were exposed to glutathione, but two different strains were used. A Park-RVA strain in which the parkin protein is fully expressed. Also, the Park-25 strain was used, which does not allow the expression of the parkin protein. The parkin protein is another protein linked with the cause of the development of Parkinson's disease. With parkin, only one set of dopamine receptors was found to have a significant increase of the neuron toxicity compared to the group with no parkin. This shows that glutathione lessens the effects of parkin due to its antioxidant properties. Based on the research of Whitworth, Congdon, and Gruber, it would be expected that the higher concentrations of methyl blue, mitoQ, and glutathione used in this experiment will significantly reduce the production of alpha-synuclein in C. elegans, while lower concentrations of these chemicals will have little effect on the reduction of alpha-synuclein. The purpose of this experiment is to see if methyl blue, mitoQ, and glutathione reduce the production of alpha-synuclein. In this experiment, there will be 8 different groups. In all of these groups, the C. elegans will be from the OW13 strain, which makes the C. elegans produce alpha-synuclein. The groups include a positive control, a negative control, 6 microgram per milliliter methyl blue group, 60 microgram per milliliter methyl blue group, 10 milligram per milliliter mitoQ group, 50 milligram per milliliter mitoQ group, 10 microgram per milliliter glutathione group, and 50 microgram per milliliter glutathione group. All of these groups will be exposed to heavy metal stress using lead nitrate, except the positive control. The LD50 of this stress will be found in the pilot study. To keep the worms in each group age synchronized, they will be induced under a chemical named FUDR, which inhibits the C. elegans from reproducing. The production of alpha-synuclein will be measured using a YFP assay, and the longevity and locomotion of the worms will also be measured. The data will then be analyzed using an ANOVA followed by a post-hoc Scheffé test. This project is doable as long as all of the correct safety procedures are taken when dealing with heavy metals that are being used in this experiment. When lead nitrate is being handled, the user will wear lab apron, goggles, and gloves. When creating agar plates with chemicals, gloves, goggles, and a lab apron will also be worn to ensure the safety of the researchers. All of the materials are not expensive except for the yellow fluorescent filter. The yellow fluorescent filter can be used for many projects in many years to come, not only for this project. OW13 C. elegans will be used in this experiment and will be purchased from the CGC. Methyl blue, mitoQ, and glutathione will need to be purchased since they are the variables used in this experiment. A fluorescent microscope is needed for this experiment, so the amount of alpha-synuclein produced by the C. elegans can be analyzed using a YFP. For this YFP, a YFP filter will be needed to be purchased for the needed wavelengths. Another microscope will be needed to observe the worms. FUDR will be used to inhibit reproduction in the C. elegans for the age synchronization protocol. Lead-2 nitrate will be used as the heavy metal stressor in this experiment. Nematode growth agar will be needed for the C. elegans to live on. Also, Petri plates will be needed to hold the agar. The E. coli OP50 are needed since E. coli act as the food for the C. elegans. M9 buffer will be needed to transport entire populations of C. elegans from plate to plate. Micropipettes will be needed to correctly measure the concentration of the variables.